in February, I took a trip to California, and on the West Coast airports in California, I saw people wearing masks, and I said, this has to be serious. I've never seen this before. And that's when I just started saying, okay, let's figure out what's going on. I'm going to start at the very beginning. I'm going to look at the papers that discovered the virus and, take it wherever it leads me. And I never expected to find that those papers didn't actually show any virus. I was really surprised that the experimental techniques that they were using, it couldn't possibly show a new organism. It was totally the wrong experiment to do. At first I was just confused. I'm like, I must be missing something, because why would it be so far off? It was just shocking, and I had to really talk to other people and look at their work and look at the historical context before I could fully appreciate what was going on.
With any virus, to show that it exists, you need to separate it entirely from any biological material.
And the studies, and the most important studies, claiming that they have proven SARS-CoV-2, they haven't purified the particles. And without purification and finally isolation, you cannot say that you have found a virus.
When somebody says, a virologist says, we have isolated a virus, I thought, as does everybody, that they took, 100 people or 500 people who are sick in the same way, right? They have a runny nose or a cough or pneumonia, and then you do steps, like you take their secretions, you then macerate it, and then you filter it, and then you centrifuge it, and then you have a band that has just those viruses, right? That's what everybody thinks they did. And then you tell people the steps, so some other lab could repeat the steps. That's how you do it in science. And then everybody says, yes, this thing that we have is pure SARS-CoV-2 coronavirus. That's what every human being understands by the word isolation, and that's what every doctor and human being thinks happened in this case, right? Except it didn't happen once.
This is the story of a group of doctors and scientists who are challenging the very heart of the scientific foundation that formed the pandemic. They are iconoclasts, scientific dissidents, pointing out crucial flaws in the very experiments that claim to have discovered SARS-CoV-2, and they are profoundly at odds with medical authorities. Yet, they are part of a long tradition of doctors, journalists, and scientists who have spoken out against the validity of the experiments used to show that one particular disease or another was caused by a newfound virus, but who have time and time again been written out of medical history. It's a scientific fight that goes back more than 100 years, beginning with a debate between two chemists, Louis Pasteur and Antoine Béchamp. But its modern incarnation began as a disagreement in 1985 between on-the-ground AIDS researchers and none other than a young medical bureaucrat named Anthony Fauci.
It was declared Well, HIV, the AIDS thing, April 23rd, 1984, before any scientist or anybody else in the world knew there was a AIDS with a capital AIDS, contagious infectious disease, nobody in the world, no scientific paper was published or anything about it. It was just declared on that day, and it became government policy, and it's still government policy to this day. That set the stage. Not only that, it set the stage with having a disease with a virus that has never been obtained from a human being. This HIV thing, the only place that I know of that it exists is in the laboratory. Nobody has ever published obtaining or isolating or even seeing HIV in a human being.
The young Fauci had thrust into the mainstream the ideas of cancer researcher Robert Gallo, that a particle he had claimed to have discovered, the HIV virus, was the cause of AIDS, that this virus could be sexually transmitted, and thus AIDS was at the very edge of becoming a new global plague. While much of the world rushed to congratulate the NIH, a small group of doctors and scientists pointed out that the declaration had no serious scientific backing, and that the so-called HIV virus had, in reality, never even been proven to exist.
I was here living in San Francisco. I had my own little biotech companies. So I wanted to work on this like everybody else in the Bay Area wanted to work on AIDS. We wanted to put our skills to work to try to cure it. Be heroes. Be carried on people's shoulders, like all young scientists. We talked about AIDS from the very beginning when it popped up in the 80s, even before it had a name. And then here it was in 84, it's a virus and everything. Oh, good. I'm a chemist. I'm going to make inhibitors to kill this retrovirus. And so the other people working here and there. And then by 87, I've been starting to question this stuff, 85. But between 85 and 87, I started talking about the problems with this whole notion that AIDS is contagious and caused by a transmissible infectious agent. All those anomalies, One of the first things that made me suspicious back, I think it was 84, 85 for sure, about this HIV business was, how does this virus know that you're gay and I'm supposed to cause Kaposi sarcoma in you and not in these IV drug-using white men? I give them TB or something like that, I mean, that was one of my first suspicions, And also, how does that little virus know not to cross the street? Like in the Castro area. Stay live here where the gay guys are, where they have all these AIDS diseases, but it doesn't go over here in this other part of town. It's a virus that knows your zip code, whether you're gay or straight, white or black, or whether you're rich or poor. That's one of the first things, that those discrepancies was back, by 1987, I realized this whole shoot match with contagious sexually transmitted HIV was not tenable. In fact, it was crap.
The debate quickly blew up when the leading virologist in the U.S., Peter Duesberg, accepted the government's claim that what was pictured under the microscope was a so-called virus, but published a paper explaining in 19 clear points why this particle could not be the cause of AIDS.
It is dormant to begin with. It's dormant when you die from it. It's dormant when you suffer from it. There's no report in the literature describing the virus ever to be active in a patient.
It was a silent war waged behind the scenes. As the debate shot through the scientific community, the critics were soon joined by Kary Mullis, the Nobel Prize-winning inventor of the PCR process.
And at some point we needed to re-up our grant from the NIH to work on that, and I had to write it. And so the first line of that was, HIV is the probable cause of AIDS. And I wrote that, and then I said, well, I need a paper, some kind of scientific paper, to reference that statement. Because when you make a statement like that, that's like a fact. You need to say, here's how come I know that. Right? You put a little 1 if it's the first statement you've made, and then you put down at the bottom of the paper you have a 1, and you say, here's a paper by somebody that describes why that statement's true. Right? And so I said, well, what's that? I don't know. Let me think about, what is that paper? Who do I go to for that? And I looked around. I asked a couple of virologists at that company, and they said, no, you don't have to reference. I said, I have to reference that because I don't know where that came from. How do I know that? And it turned out that nobody knew it. There wasn't a scientific reference, like a paper that somebody had submitted with like experimental data in it and like logical discussion and said, here's how come we know that HIV is the probable cause of AIDS. There was nothing out there like that. Nothing.
Mullis insisted his technique was being misused to claim it could find viruses and diagnose disease, when it could do neither.
AR is separate from that. It's just a process that's used to make a whole lot of something out of something. That's what it is. But it's not. It doesn't tell you that you're sick, and it doesn't tell you that the thing you ended up with really was going to hurt you or anything like that.
Mullis became Fauci's most prominent critic, and soon was joined by numerous other leading scientists, but it was a debate that was kept almost entirely out of the media.
To say that Anthony Fauci controls the media is a real understatement. Here's a story Peter Duesberg used to tell. Peter was going to be on one of the talk shows. I don't know if it was CNN or if it was one of the big, I think it was actually one of the networks, and they were just going to do straightforward journalism. Oh, Dr. Duesberg, you are an esteemed retrovirologist. Why don't you think HIV is the cause of AIDS? Why everybody disagrees with you, but what do you have to say? So they flew into New York. He was in his hotel room and about to go downstairs to get picked up by the car, and phone rings. Oh, sorry, Dr. Duesberg, we have to cancel the interview. And he says he literally turned on the TV at the appointed time, and Tony Fauci was sitting there talking about the latest numbers and the latest scare and how. So they must have had such a surveillance capacity even then, and or spies and moles and all this kind of stuff, so they absolutely knew when somebody was going to interview Peter Duesberg.
The argument came to its most devastating culmination over Fauci's prescription that those who had tested positive for the HIV virus, using Kary Mullis's PCR test, should take a repurposed chemotherapy drug known as AZT to save their lives, while Fauci's detractors argued that the drug was a known and extraordinarily deadly poison, which was killing the very people who thought they were saving themselves. It was an argument over a new disease, but not a new argument. 30 years earlier, a number of prominent doctors had spoken out forcefully that polio too had been wildly misdiagnosed, and that the devastation of childhood paralysis was much more likely due to the introduction of new insecticides than to the sudden and capricious rise of a new virus. They too were silenced by a growing medical bureaucracy headquartered at NIH under the influence of the now dominant post-World War II pharmaceutical companies. Even earlier, a silent war had been waged in the medical community over smallpox, which had come to a head when the very doctor who had been selected to write the Encyclopedia Britannica entry for smallpox vaccination set about to look at the research and found, to his shock, that vaccinations had clearly not been the reason smallpox had declined. Though considered the most prominent British doctor of his day, his entry was deleted, his career attacked, and his research written out of orthodox medical history. The thrust of these doctors' and scientists' arguments is that a number of profoundly unscientific experiments had emerged, which were being used to place the blame for disease on the concept of tiny, unseeable entities named viruses, which could not in reality be found in sick patients, when the real causes of disease, such as smallpox, polio, and AIDS, were industrial contaminants, toxins that emerge from poor living conditions, malnutrition, and new pharmaceutical products that were destroying the body in untold new ways. Yet despite their efforts, a rapidly growing edifice of bureaucracy was forming, built on the foundation of virus theory, and the idea of viruses as the cause of disease permeating the culture so profoundly that slowly most doctors came to believe in it. The debate has reemerged today, as a growing group of doctors and scientists are speaking out against the experiments that form the justification for the SARS-CoV-2 pandemic. Their argument is that while thousands of experiments have been done regarding the SARS-CoV-2 virus, they are built on an unscientific foundation that is, in reality, pseudoscience. Today, medical authorities have placed virology center stage in claiming it has the answers to human sickness, and that the rapidly mutating deadly SARS-CoV-2 virus must be controlled through urgently necessary population-wide medical experiments and the rewriting of the world's long-standing social contracts. But the argument of their detractors is that this story has become transparently false, that the experiments to justify these extraordinary actions, when looked at closely, are unscientific and contradictory, have not prevented, but caused death, disease, and mayhem on an enormous scale, and must be brought to an immediate end before more devastation ensues. The argument from the mainstream view is well known, but the arguments of its detractors have once again been almost entirely silenced. This is their story. We all remember when the pandemic began, on the pronouncements that a new, deadly, and highly transmissible virus had been found. The claims were based on a single paper out of a laboratory in Wuhan. But while much of the world set into panic, a number of doctors and scientists were examining this paper, and the subsequent ones, in detail. Here's what they found.
The first thing I did, I actually went to look for the first Chinese study from the CDC, CCDC, the Chinese Center for Disease Control, published under the name Zhu and others, which was the first supposed isolation of the SARS-CoV-2, and of course immediately found incredible discrepancies and a methodology that didn't isolate anything.
They found some people with atypical pneumonia, which is not uncommon. Every year we see lots of atypical pneumonia. That's what we often call, the flu or a virus, because we don't really know what's causing it, but, every winter you see this. So I was like, well, they see a few cases of atypical pneumonia. That's not unusual. They had some commonality that they were all at this outdoor food market, which didn't have the best sanitary conditions. So I'm like, if people got sick from there, I would think some kind of food poisoning. But they never even came up with that idea. They said, it must be something like SARS. And it was just, why would they come to that conclusion? And then this was also the weird thing, because if you really do have a new disease, right, you would want to take a good sample of cases to study, like 1,000 people, or at least 100. They had less than 10. Why would they only study such a tiny number of people? And I think they only actually took samples for these tissue culture experiments from 3 patients. And then all of a sudden they're assuming it's like some deadly virus plague.
So-called isolation or discovery of this virus is done in a single individual, a single human being. All right. There were 44 cases of pneumonia in December, I guess it was, of 2019. And the World Health Organization office there in China cataloged these 44 people of pneumonia that they had no explanation for. And this is in a country that has about 1 million pneumonias every year. All right. What is so important about 44 cases? They say don't know what's causing it. Lots of things can cause pneumonia. Chemicals can. Malnutrition can. Drugs can. Being old can. Being in a hospital can. Chronic diseases. There's all kinds of things that can cause pneumonia. And with this one guy, so now they have this one guy in a country of over 1 billion people.
So essentially, what I found was that they were taking the liquid from patients, and they were maybe centrifuging them or filtering them, but I made a calculation that in the 15 micrograms that they were using to do the test, they were still like about 30 billion particles, virus-like particles, of which 85 to 95% of human origin, human genetic origin, and they would take this very heterogeneous material and put it into a culture, and then they would say that that was the isolated virus, which of course didn't make any sense.
They're taking samples from the patient to try to find a virus, but they don't look in that fluid from the patient for the virus. I'm like, well, why not? That's the source of it. Instead, I see they mix it in this foreign cell culture of monkey cells. Like, why would they do that? It doesn't make any sense at all. They basically combined the words virus and isolation together and created a new definition for that term. And this is why it's so confusing. So only the scientists or doctors who actually take the time to read the paper and read the methods are going to understand that there's no isolation going on. Everyone else who just reads the title of the paper, they'll just, they said it right here. They said isolated.
They don't actually isolate viruses. They just say that they isolate viruses. If you think of something like a bacterial species, and it is possible to isolate bacteria, as in just get the bacterial cells, separate them out from everything else, and then analyze them, work out what their composition is, their genetics, their structure, work out what the bacteria, how they behave, what they can do, etc. And I think most people think that with viruses, they're doing the same thing, except that with viruses, when they use the word isolation, it doesn't mean that they've physically separated and purified the particles. It means something totally different. So that's the first deception.
So the question is, why don't they do it? Why don't they show us this virus from a sick person? Not only in 100 people, but in one person. So I don't know the answer to that, but what they say is, well, there's not enough of these virus particles in a sick person to find them. Which is odd, because if there's not enough viruses in a sick person to find them, what's the theory that they're going to make us all sick and kill us all? So that's obviously ridiculous.
In the virology papers that form the foundation of the pandemic, indeed, the term isolation is used in an entirely different way from its ordinary usage. What the papers mean by the term isolation is that the nasal or throat swab has been isolated from the patient, not that the virus itself has been isolated from the nasal swab. Without isolating the hypothetical virus, these doctors and scientists argue that no independent variable can be properly studied or characterized. It is the first step in what is considered the bedrock of both the scientific method and the founding principles of microbiology, known as Koch's postulates. Through FOIA requests, an activist named Christine Massey has received confirmation from more than 150 government institutions, including the CDC and NIH, that they have no record of a virus named SARS-CoV-2 ever having been isolated directly from a patient. Instead, these institutions point to scientific papers which use three methods for claiming they have discovered the existence of the virus without ever having isolated a virus itself. The first of these is known as the cytopathic effect.
People have sent me in this past year, let me just take a guess, 50 papers called The Isolation of SARS-CoV-2. Now, maybe 48, but you know what I mean, it's a lot. And both the normal virology community and the sort of alternative community sends me paper after paper saying, here's how they isolated, here's a paper showing the isolation of this virus. Every single one did it the exact same way. They take snot and they inoculate it onto tissue culture, usually monkey kidney cells, which are called vero cells.
And put it in this recipe of the toxic cell culture with, toxic antibiotics, and they usually use ones that are specifically toxic to the type of cells in the culture. So when they use kidney cells, they use antibiotics like streptomycin, gentamicin, and amphotericin B, which are all specifically toxic to the kidneys. They starve the culture, and then they add other biological materials like bovine calf serum, like a fetal bovine serum, rather, like from, calves in the womb, their blood. And they can show these, what they call cytopathic effects, which is damage to the cells from this every single time, and then when cells are damaged, they make particles like exosomes and apoptotic bodies and other types of particles that don't even have names. And they can just point to whatever particle they want and say, there's the virus. So it's very convenient. It just doesn't come close to proving what they say it does.
The way that they prove, inverted commas, SARS-CoV-2 exists is through stressing cells in cell cultures, vero cell cultures. They stress them with antibiotics, all sorts of mechanisms, and then they see things bud, or they see cilia on the cells stop beating. And so they interpret those as virally caused, yeah? Or as viruses budding, or as the cilia on the cells, the human cells, they stop beating, so the cell must have died. But it could be due to all sorts of things. Very, very uncontrolled experiments, one-shot experiments.
The cytopathic effects are just non-specific effects that occur in a test tube when you put